Okay, I'd like to welcome everyone to the second meeting of the IMAG MSM Working Group on Multiscale Modeling for Viral Pandemics, our mini seminar, and our discussion. The agenda today is going to consist of a brief introduction, a mini seminar by TJ Sego of Indiana University, and then a discussion of the status of the working group. And again, I see people are signing in at the moment, so maybe I should give them another minute or two to get in. So just to remind you that currently Reinhard Labenbacher and I are co-leads for the working group. We have Jim Sluka, who is working on the web side of things and also can help with technical issues. And as activities coordinator, Bruce Shapiro, who's been generously sending out emails and coordinating with people. So if you have any questions or comments, suggestions, we're all available to discuss any issues that you find important. We have a Slack channel. I encourage people to, perhaps, Jim, you could paste the Slack channel link into the chat, if you would. I encourage people to use the Slack channel. Different people have different favorite means of communication. There was a suggestion that there should be a weekly or multi-weekly topic for discussion. That's a great idea, but it does need people to essentially volunteer and kick off for those activities. And I'd encourage people who are interested in being leaders on the Slack, especially people who are leaders of subgroups, to speak up and help get the Slack going. In particular, if we're going to have weekly or monthly discussion topics on the Slack, we probably need somebody to coordinate to get that going. The first week or two, we had quite a bit of Slack activity. This past week, not so much. It's a communication channel like any other. It is useful if we use it. It's less useful if we don't. So that was my main introductory material. Today we have our mini seminar by TJ Sego of Indiana University, who's going to talk about model sharing and modularity in the construction of multiscale models of viral infection and immune response. And this is going to be a short mini seminar, in the order of 15 minutes or so, with some time for discussion. We only have one talk scheduled this week. We'll come back to the schedule for subsequent weeks after the talk. If you would like to talk, please let us know. We'd like to get you on the schedule. This is not a COVID specific working group, and therefore any work that you do with respect to immune response, the immune system, viral infection, or other kinds of infection is appropriate for presentation. Also, experimental work that's relevant to modeling or modeling infrastructure conversations are also appropriate. So please don't feel that you have to have a COVID talk in order to be somebody who speaks for us. So with that, I'd like to turn it over to TJ. So I will unshare. TJ, you now have, I should be able to, let me make you co-host here. You should be able to screen share now and take it away.
Great. Thank you, James. Thank you all for being here today. I'm very excited to be here and talk with all of you about the work that we've been doing out of IU on developing a modeling and simulation framework for multiscale, multicellular spatiotemporal modeling of viral infection and the immune response. So to start, just a little bit about me. I'm a postdoc working with James at IU in the Department of Intelligent Systems Engineering. I did my PhD at Purdue in mechanical engineering and finished that last year. So I just started the second year of my postdoc. And my research tends to revolve around all things computational modeling, computational mechanics, biology and biomechanics, so on and so forth. My primary research working with James, specifically in computational biology and cell biology in particular, which is what brings us here today. This talk is based around this simulation framework. I'm going to try to drop a couple of links into the chat to be able to quickly access some of the information that's on the screen here. So we have the first paper about this framework that we've constructed. This is a multicellular spatiotemporal modeling and simulation framework built in CompuCell 3D, which we develop and maintain in Bloomington at IU. There's the chat. So I'm going to drop some of these links into the chat so you can quickly grab those. So the paper has been preliminarily accepted with PLOS-Comp Bio, so I can't hand you a published version of the paper right now, but the link to the preprint is what was submitted on acceptance, so you can find the modeling details, most of which we'll sort of gloss over today to focus more on the modeling aspects concerning modularity and model sharing and so on and so forth. You can also try the simulation framework. You can see some screenshots of it down here at the bottom. You can try the simulation framework with our NanoHub tool, which is the second link that I've dropped into the chat. So this is a web-based platform, so there's no installation or anything like that on your computer. You can just follow that link, do a quick registration, and run the simulation yourself from your browser of choice. And then for those who are more inclined to the programming side, this simulation framework, along with everything else CompuCell, is open source and available on GitHub, so you can find the source code for all of the modeling work in its current state, as well as its entire state throughout development at the third link that's been put in the chat, which is shown right here. So without further ado, let's just dive right into it. So our modeling framework works under the basic premise of that we're going to look spatially at a local area of infection in a planar epithelial sheet. And then we're going to model what goes on during infection with spread of infection from cell to cell. As you can see here, got a little blue sheet of epithelial cells and some different colored cells showing different states of infection, so on and so forth. And then we model transport of virus and other species around that, as well as recruitment of immune signals, systemic signaling as well for organismal response, and then do lots of subcellular modeling viral kinetics inside each individual cell as well. So the basic premise that the framework is currently built on is that we, in our spatial domain, we look at a quasi two-dimensional patch of susceptible tissue. So this is modeling infection in an epithelial sheet. You can see this shown in the top row on the right. So this is a layer of epithelial cells like shown in the previous slide. And for the simulations that we've done so far in the models that we've built, we do our modeling based on the assumptions of primary infection as well as acute infection. So the models that we've developed and employ right now don't represent anything like a pre-existing adaptive immune response or any sort of antibody production or existing memory T cells. And the simulations that we've done so far have more so focused on shorter time frames on the order of a few weeks, in which case we look for either the virus to be cleared due to the immune response or total infection. Now, the models that we've built so far that we employ, a schematic is shown here on the right for all of the different models in our main framework. And we work primarily with a simplified version with two different cell classes. These are general cell classes of epithelial cells and immune cells, which can take various stages depending on what our different submodels dictate. And we represent 3 diffusive species that are simulated using reaction diffusion, so mass transport of diffusive species around our cells, specifically with an extracellular virus field. And this is released by virus-releasing cells after infection and then a subsequent eclipse phase. That virus then goes on to infect other cells due to internalization. We have infected cells that release a generic cytokine field. That cytokine field does lots of inflammatory signaling from recruitment of immune cells to the local patch, as well as guiding immune cells due to chemotactic migration. And then we have a third field that's an oxidative agent. And that oxidative agent is released by immune cells when they're sufficiently activated, that then goes on to kill epithelial cells when they're sufficiently exposed. And then we do some higher level systemic modeling using a compartmental model that represents a lymph node. So what we do is that we take the total amount of local cytokine in our spatial domain, and we use that and map that as a recruitment signal to our compartmental lymph node model that stimulates the production of immune cells that then results in later arrival of immune cells to our local tissue patch. Now, most of the model modules, I'd like to just sort of pass by and be happy to talk about those separately or in discussion and so on and so forth. There are a few of interest, I think, to sort of motivate some of the work here, though. So we have individual model modules that represent different aspects that we're interested in modeling, here shown for the modules of the viral life cycle in our model. So a schematic is shown here on the right. We have our extracellular virus field, and we have an individual epithelial cell, and we have the processes of internalization, where the extracellular virus field goes from the extracellular space into an individual cell. And that's its own model module. It has its own expressions. It takes the information of local virus and determines how much, if any, virus goes into a cell. Now, there's a second model module in the viral life cycle for modeling viral replication. And the actual equations are shown here on the bottom right. So this is a system of ordinary differential equations that model the quantities of viral genomic materials at various stages in viral replication, from unpacking through replication all the way to assembly, the final quantity of which, assembly, is then passed to another model module, which is a model of viral release. So this is taking information from viral replication, taking it out of that model module, and then releasing it back into the extracellular space. So each of these are individual models with their own individual sets of equations. They have their own implementations and so on and so forth, and they're all connected and pass information back and forth. But as far as the actual calculations that go on inside each model module, the other models don't exactly care. They just look for specific information to be passed to them and return information to whatever else they've been connected with. And this is what it looks like. So this is a typical simulation for 2 weeks. We see here in the top right, so this is a layer of epithelial cells on top of which immune cells are seated. So these are, again, recruited due to local cytokine signaling. Those immune cells roam around and do things like release an oxidative agent, which is shown here in the bottom right, that kills epithelial cells. They also do contact killing. So when they come in contact with virus-releasing cells, they kill them on contact. You can see the extracellular virus field and notice the correlation between red virus-releasing cells and increases in the extracellular virus field, and then the cytokine field that's released by infected and virus-releasing cells. So this is an animation from our simulation framework, and this is what will run if you follow the NanoHub tool. There's lots of stochasticity in these models, so your outcomes may look a little different, but when you run the NanoHub tool, this is the sort of simulation that you'll find. Now, we've done all sorts of interesting things with our framework so far, along with building additional models. I just wanted to show a brief application of this. So We, in our paper, and you can read more about this if you follow the link to the preprint, this is a schematic of a scenario of drug therapy that we simulated. You'll recall with the equations of the viral replication model that there's this second quantity, which is the amount of viral genome that's replicating. In particular, this red boxed in term is a term of exponential amplification. So this is actually the production of more virus from the existence of current virus. And specifically, to do something like modeling an RNA synthesis blocker, in the simplest case, we can just say that a constant coefficient, like what's shown here for this R max, we can just say that a coefficient like that could be affected by an RNA synthesis blocker. And this is shown here in the top right. In our drug scenarios that we've simulated, we said that at various times in simulation, we have our coefficient R max, and we say that at some moment the drug therapy is applied, and that results in a reduction in the exponential amplification of replicating viral genome. And this is what it looks like. This is a parameter sweep varying the time in simulation when the drug therapy is applied in the columns. And then by row, this is showing from top to bottom decreasing potency of therapy. So in the top row, this is a complete shutoff of exponential amplification all the way down to what's shown here with a coefficient 0.875. This is a 12.5% reduction in the coefficient Rmax. And you can see in these green shaded regions, we're looking at early, very strong therapies. And in the red region towards the bottom right, very late, very ineffective therapies. And we see that in the green regions, looking at the number of uninfected cells, we get very little, if any, infection due to something like prophylactic treatment. Whereas in the red region, we see the number of uninfected cells going to 0. In the right pane, we see lots of extracellular virus due to continued spread of infection. And then there's this sort of region in between that I like to call a fuzzy boundary, where we get lots of stochasticity. You look at the number of uninfected cells in this boundary in between, you get multiple simulation replicas running the same set of parameters in the same initial conditions, and the stochasticity and being on this sort of bifurcation in the emergent dynamics of the system, we can get drastically different outcomes from complete infection to almost no infection, even for the same parameter set. Now, as I mentioned earlier, the framework is developed in CompuCell 3D. Again, this is the simulation platform, modeling and simulation platform that we develop at the Biocomplexity Institute at Indiana University. CompuCell, again, is open source and available on GitHub. It comes with a full PDE solver suite that's completely in-house and tailored directly for the type of modeling that we do. We also distribute that with libRoadrunner, which is, to my knowledge, the fastest in class ODE solver available that can be attached to free-floating simulation domains to model something like systemic response, or attached to individual cells to model intracellular dynamics. So these are the two solver suites that were used to do viral replication as well as immune response modeling. We also distribute CompuCell with Player, which is a GUI for doing real-time interactive simulations, so starting, pausing, restarting, exporting simulations in real time, as well as Twedit, which is a code editor for doing easy model specification, which in CompuCell is done using XML and Python programming language. And in particular, CompuCell is very good about presenting the ability to do modular model specification. And we have exploited that quite a lot when developing this simulation framework. So this framework is developed with model modules, as I mentioned earlier, that are designed to be interchangeable, they're shareable, and they're extendable. Each of them individually. All models are represented in individual modules, and those can be manipulated, replaced, so on and so forth. And this resolves down to a simulation specification method that consists of loading existing model modules, whether developed by the user or by us, just loading them, giving them to CompuCell, and then telling it to run. Example of this is shown here on the bottom right. This is just importing a particular script where a specific model module is specified, registering it with CompuCell, and then telling it to run. And so what this is resolved down to for the simulation framework is that for the paper that we've written for our initial paper, we have a set of model modules that were shown in the previous simulations. We just call those the main modules, just for convention. Those are what runs when the simulation framework is just ran as is out of the box. So we call those the main modules. And then we've set up a sort of sandbox, a library of what we call add-on modules, which is a space for both us and for others, whether collaborating with us or not, to develop extensions, to develop additions, to replace models that maybe they don't like, something that we developed that they think could be improved, or extending them and adding new biological detail, adding new biological mechanisms, and so on and so forth. And this is designed specifically to prevent collisions between, say, two different groups working on different aspects of the same framework. So it's certainly possible to go through and work on the same code and overwrite work that others have done. In this case, we've specifically set up this sort of sandbox library to prevent that, so that lots of groups can be working on lots of different aspects of the framework simultaneously, all contributing to the same repository, which is available at the GitHub link that I posted at the beginning of the chat. So we generated this example sort of scenario that's shown here on the right. So one of the models that we didn't include in our main modules is a model of tissue recovery. So when a cell dies, as shown here, say on this subpanel B, we see some black areas. Now, blue in these simulations represents uninfected cells, and black represents dead ones. And so a patch of dead cells like this would be something like tissue damage. And what we did not include in our main modules was recovery of that damage. What to do with the dead cells when the virus is cleared and the tissue goes back to homeostasis. So the scenario that we contrived was to first begin by imagining that one group decides that they're just going to create a simple recovery model where the dead cells just turn back into uninfected cells with a simple probability. It's a discrete stochastic version of just a continuous recovery rate of dead cells converting back into uninfected cells. And that's shown here on the top right. On the next slide, we're actually going to look at what this looks like. But just glancing at this now, this simple model is just very basic dead cells stochastically converting back into uninfected cells. And then we imagine that a second group comes along and they say that they like that basic idea. They like the mechanism of recovery, that uninfected cells show back up from the presence of dead cells, but they would like to add some details to the actual processes of recovery and make those something like dependent on the neighborhood of the dead cells. So making the tissue recovery depend on the consistency of the neighborhood of a dead cell. And we imagine what would be the easiest way for a second group to come along and improve upon the work of the first group. Now, the fast and dirty method is to copy and paste the source code developed by the first group, but instead what we've done is we've built in the support so that another group can come along and instead just import the source code originally developed by the first group and instead change or extend or even just replace aspects of it using basic Python programming features. We're gonna look here very quickly. Don't pay attention too much to the source code, but rather more the annotations of what's shown here. So here on this left column, this is group A, and this is the simple recovery model. So this is just going through in every simulation step, just looking at every dead cell and asking, according to our recovery criteria, does recovery occur? And if it does, what happens during recovery? And that is the entire model specification. This is exactly what is shown in the previous slide for the simple model of recovery. Not very much work, but that's a feature of CompuCell. The feature of the simulation framework is what happens with Group B. Group B comes along and says, we like testing for recovery at every simulation step, and we like what happens when recovery occurs. But what we would like to improve upon is the recovery criterion. And so what this looks like is Group A can create a model module in the add-on library directory. So the simulation framework has a built-in directory to contain all add-on modules in a simple directory called models. And in models, there is a uniquely named folder for each model module developed by either us or someone else. And inside that model module is where all of the code is defined for the model that's implemented in the model module. So in this case, for Group A, they have their simple recovery model. They decided to name their module Recovery Simple, and they just have this tiny bit of code in this file called Recovery Steppable. Now, Group B comes along and they say they want to change the recovery criterion. And so just exploiting the architecture that's been designed for this framework and basic Python programming, they just just grab the source code and import it into their own script that's defined in their own module. Their module is called Recovery Neighbor. And in Recovery Neighbor, they have their own code, their own file called Recovery Steppables. And in Recovery Steppables, all they do is they just import what was previously defined by Group A, and then they just overwrite the criterion. And that's it. And so when Group A, Group B, or Group C, some other group, or us, we then can come back to the simulation framework and we can use those models as well. We can come back to our main simulation script where we import our main model modules, in this case shown here in the bottom right. This is at the very end of importing all of the main model modules, this one for modeling the effects of the oxidative agent. And then you'll notice the next two lines, importing the neighbor recovery steppable. This could be Group B's code, this could be Group A's code, this could be someone else. They can select whatever models and modules have been developed by what other groups happen to be contributing to the framework, whether collectively or even in competition. Now this can get as complicated as you like, dare I say as complicated as biology gets, but we've gone through and test-driven this a bit more to show some basic ideas about the general scope of the framework. In particular, as what's shown here, this framework was developed for SARS-CoV-2, but the computing infrastructure is not particular to SARS-CoV-2. So the main framework that will run when you launch the NanoHub tool will use a viral replication model that was developed for and calibrated to SARS-CoV-2 data. However, we do lots of work maintaining and improving this framework to continue to support modularity of this framework for applications outside of SARS-CoV-2 particularization. What's shown here is the ability to very easily, in exactly one line of code, replace the viral replication model that's being employed and used in every individual cell with a different viral replication model altogether. In this case, what's shown here, and this is also described in the paper, so this is a replication model developed by Alan Perelson and some of his collaborators for hepatitis C. So not SARS-CoV-2 specific at all, but rather just a complete substitution of the viral replication that's modeled inside each individual cell. Otherwise, everything else is unchanged. And then we run it again, and we get, interestingly, some different dynamics. We don't explore this too much in the paper because we were specifically interested in SARS-CoV-2 for the actual modeling applications, but the computing infrastructure is there to use this in a general sense to incorporate lots of viral replication modeling that's been done in previous work or with ongoing work that's developed even now. So I just want to finish up very quickly by showing some work that we've been working on that I'm particularly excited about. So this is a project that some of us at IU are working on with some of our friends in Pittsburgh, specifically with Professors Ericka Mochan at Carlow, as well as Bart Ermentrout at Pitt, both of whom may be here. I was talking with Ericka earlier. So Bart and Ericka developed a very nice ODE model of influenza infection and innate and adaptive immune response. Now the figures are shown here. These are all taken from the paper that describes this. This is cited here at the bottom. Ericka was kind enough to provide all of these figures, and I'm very grateful to her for having done that. These are excellent figures describing a very complicated model, just shown here in the top right. This is 20 ODE equations describing infection dynamics and immune response, everything from innate response like neutrophils all the way to B cells, antibody production, type 1 interferon, so on and so forth. So one of our projects has been to generate a spatial analog, a spatial version of that ODE model of influenza infection, and then take that those models, the implementation of those immune response models, and then plug them into the simulation framework as an optional model module. So this is my sort of favorite project that I've been working on right now, and this is what it looks like. So this is the same premise except with lots more immune cell types, lots more fields, so on and so forth. So the immune cells are shown here in the bottom left. You can see there are a lot more types there, epithelial cells with infection, a much bigger domain, more signals, and so on and so forth. So with that, I'd like to open it up for questions. James, if you had anything you'd like to mention, you've been involved with quite a lot of the work that's been shown here today. So if you had any comments that you'd like to make as well.
No, please. I think Jacob had a question. You should be able to unmute yourself.
Yeah. So I saw that you use Python code and you are able to replace either Python code. This is quite regular. However, one of the issues is that, especially if you, with Python, when you write something good, you include many libraries that are external. So how do you cope with this? Is it the responsibility of whoever it is plugging into model to plug them in, or are you using other mechanisms like Docker to actually go and wrap everything up and give it to you?
Yeah, so that's a pretty advanced, that's pretty far down the line. And we have put some mind towards that, but have not gone as far as to work on those aspects, which is important, especially depending on what's actually being modeled. Most of CompuCell, for example, is in C++, and then we wrap it with Swig. So we put all the computing work on C++ and we do the model specification and so on and so forth in Python. The trouble with doing that functionality for community-driven development is the amount of work required to potentially support a broad community. So probably, at the very end of that tunnel, what would be required is for those who develop those modules, the burden would probably, at least for now, be on them to develop those deployments.
So basically the deployments exist in Python specifically. You can actually ask that it will be available as that whatever is plugged in is provided as a package. And there are multiple packaging tools. So like Conda, pip, or you can actually ask that the environment should be provided as Docker. And this should be some of the things that you have to require to make the environment kind of standardized.
Yeah, absolutely. I mean, in the overwhelming majority of cases, and James, please correct me if I'm wrong, in the overwhelming majority of cases where CompuCell is used, the model specification by the user is exclusively done in Python using the built-in support that we include with CompuCell. It's extremely rare for anyone to go that far into the actual computing capabilities, with the exception of perhaps developing new C++ plugins for the CompuCell engine.
Okay, we maybe the rest, that's a great topic, which we could have in the Slack and for further discussion. Are there any other questions before we move on to perhaps less scientifically interesting but necessary administrative discussion? Please feel free to raise your hand or type. If you'd rather type the question in the chat, that's fine too. Anyone?
I may have just one question
Please.
on all these models. Do you have on your repository also a site where we can find, I'm assuming you validate all these models with some kind of experimental input. Is there a list based on a specific application of what kind of data you use to inform your model?
Yeah, so for the initial paper, the bulk of the calibration and validation went towards doing viral replication modeling, specifically because we're looking at modeling SARS-CoV-2 at the beginning of the year when data was especially scarce. So for what's on the repository right now, the work has been towards viral replication, and the data sources are cited in the paper. And if I remember correctly, it's also documented in the repository. If it's not, then it will be. One of the sort of internal projects we've been working on is the repository standards. What documentation, supporting documentation needs to be included for these model modules, whether for the main modules aside from what's documented in the paper, but probably more importantly, what goes into the add-on module library, what sort of information like validation data should be defined for each of those models.
Okay, any other questions? Mamadou? Please go ahead.
Okay, thank you. For a virus, how do you really detect that molecular information and integrate it into this model? Is it like by fitting different coefficients? Can you import? I mean, how do you do that whole multi-scale integration strategy?
Yeah, it can be a pretty painful process. Some of it really relies on some of the philosophy of complex dynamics, that we describe these systems by describing their constituent elements, in which case we really focus on calibrating individual model components and then put those calibrated model components back into the complex environment. In other cases, when trying to make them all work together, it becomes much more complicated because these systems don't exactly have analytic solutions, in which case we rely on things like our HPC infrastructure. So we do lots of parameter sweeps, lots of simulation replicas, looking for things like bifurcations, trying to characterize the parameter space, which can be very difficult for a framework like this, which, if my count is correct, is around 90 parameters. Now, some of those are a bit inconsequential, while others tend to be pretty critical. But like for example, at times, we resolve down to trying to define our parameter space in terms of outcomes that we know exist when there's no availability of data that we would need. And that would be a case like this.
Okay, and any other, we've used a fair amount of time. If there's another, is there another question? Does somebody else have?
Yeah, I have a quick question. So do you think, or maybe you can comment on how this can be used, for instance, to understand why COVID pathogenicity is so different, say, from other similar coronaviruses, without refitting all the parameters in the model, just focusing, say, on one or two places where we know the differences exist, like in binding or in the interaction with the immune system?
Yeah, so the strategy that I would recommend would be to begin by calibrating the framework to well-established data. say if you're curious as to why SARS-CoV-2 is so much different from classic SARS, then start by calibrating the framework to the classic SARS data, and then testing differences in individual mechanisms and looking for similarities in the system response computationally to what seems to be the response clinically. In probably the simplest case, you could look for a very small set of variations and a limited number of mechanisms. Probably more realistically, what that activity would result in would be testing hypotheses, biological hypotheses about what the potential mechanisms are computationally. Because there are so many model parameters that, the problem then sort of comes back to the biology of asking the biological hypothetical questions and then trying to answer them computationally.
Okay. If there are no more, is there any other very urgent question? We still need a few minutes to discuss administrative. Please feel free. TJ, why don't you put your email in the chat so if people would like to talk to you offline, you'd be welcome. And now I'm going to go back to the to our agenda. We may not get through the whole agenda today. Thank you, TJ, for the seminar. I'm glad there were so many engaged questions. I appreciate that.
Thank you again.
Feel free to talk on the Slack and by email with these issues. First, provisional schedule for our upcoming mini seminars. Alan Perlson and Fred Adler have agreed to talk next week. These schedules are always tentative. Gary Ann was going to talk this week and he's on surgical duty, so he couldn't talk. So there may be slight changes. We won't meet on Thanksgiving Day. I hope people don't feel deprived if they don't have a seminar Thanksgiving Day. If you want one, we can ask. And then the week of December 3rd, we have Jacob Barak and Gary Ann speaking. And then Amber Smith scheduled to talk about her influenza work, experimental and computational work on the 10th of December, and Grace Pung from NIBIB, of course, one of the leader of IMEG MSN, has agreed to address us as well. Again, these schedule may change slightly, but that's what we have so far. Please let us know if you would like to speak or if you have suggestions for speakers. We would very, very much appreciate it. Again, almost any topic related to the immune system, model sharing, viral infection or other kinds of infection would be appropriate. And it could be experimental. Experimental data sources and data integration are also highly of interest, and the social aspects as well. We were going to come back to the current issue of the subgroup topics and lead volunteers. I want to turn that back over to Reinhardt to steer that discussion. Before that, I just wanted to turn it over for a second to John Rice, who had a comment to make on the Slack issues. So John, do you want to say something quickly about Slack channels and discussions?
Hello.
Hello.
This is Sean. My hearing is I heard my name, but am I invited to speak now?
Please, you said you wanted to talk about the Slack forum.
Okay. And this is kind of a philosophical thing about groups and group dynamics. The creation of a working group of this scope, this magnitude, breadth of backgrounds, using all virtual methodology to do it, where we're not having face-to-face meetings, poses some interesting difficulties, but I think also some advantages. And if we look at the problem we're facing, what comes to mind is the idea that what do we know, what do we not know, and what don't we know that we don't know? And it seems to me it would be very helpful to find ways to keep this group communicating around that. And that one way to do that is if every one of you wrote 4 pressing questions right now, things that you're working on, just simple questions. It's like, what we need to know is this. And we took those 400 questions because we have 230 people signed up. And we start answering them. I think that we might be able to discover some of the things that we didn't know. Because when you're looking at those questions and you have other members of the group trying to respond just informally, at that point, we begin to find out, what do we, capital W-E, the group, know? What is the body of knowledge that we share together that we have not yet shared to each other? So I just want to encourage this idea of pursuing the submission of questions, not just the question of the week idea, which is another, a good idea because we find something that's worth taking a half hour to actually talk about together. But just, would you all be willing to submit a page that had 4 short paragraphs that says, here's what I think we need to know, or this is something I need to know for work that I'm doing right now, and find out that whether collaboratively, maybe we do have a lot more answers among us than we realize. And that's the end. Thank you.
Okay, thank you very much. That's an excellent suggestion. We can certainly poll the membership and ask for a list of what you think the 4 most urgent problems facing your research are. I think that's a very good suggestion, and we'll put that.
Yep, and they can be, small questions, small, simple things directly related to something you're doing, that your model's being held up because you don't have an answer to some question. And instead of going out and trying to reinvent it, maybe somebody in the group of 130 of us knows the answer. Okay.
And Jacob, you suggested putting it in a shared document. We certainly, if we get enough responses, we had a shared document that no one used, a little bit like the Slack at the moment. And so if there are enough responses, we'll definitely do that. That's a good idea. So now Reinhard.
Produce an interesting publication to go out in 6 months. And some of the stuff that Jim is doing with the analysis of natural language in the documents that you've already submitted about your work and yourselves, applying some of that to a list of a big bunch of questions could actually maybe lead to a nice paper that says, here's the questions the group has that nobody in the group knows an answer to.
I think that's an excellent idea. Thank you. Appreciate it. And we'll definitely send out a poll on that topic. So Reinhard, one of our priorities is to try to get the subgroups organized and leads appointed for them so we can get the administrative structures put together. Reinhard, I'll turn it over to you. If you want me to walk through the list of existing leads that's available, if you'd rather screen share that on your end, that's also fine. You tell me how you'd like to run it.
Well, so first of all, I should say I have to leave in 9 minutes. I have a hard stop at 4 o'clock. But let me just say, so this is a lot of people we have in this working group, which is wonderful. It's in excess of, I think, maybe 130 or 140. And so a major challenge is to organize this group in a productive way. And of course, the subgroups are a key organizational principle. When we started this process, James and I made up a tentative list of subgroups, and then as people joined, subgroups were added. Others, look like they're orphaned. And here's James is putting up a list. And what we were hoping is that this would be sort of an emergent property kind of feature that people who had interest in particular subgroups would would sort of reorganize, merge, split different subgroups. Some of that has happened. Some of that I think is still needed. We have some people that have agreed to lead certain subgroups. Some subgroups have several people as leads, others don't have any people as leads. I think we'll pretty soon we will need to sort of finalize an initial list of subgroups with leads. So if there's any interest you have in participating in this organization, we very much encourage you to look at this list of subgroups, volunteer as a lead or co-lead for some of them, or propose a new one that you think is not covered by the existing ones. And so then the question is, what should these subgroups do in terms of organization? And let me give a more sort of a pedestrian version of what John was just explaining. In my mind, I think the subgroups sort of should, in one form or another, do two things. One is, as John said, make a survey of what we know with respect to a particular subgroup topic, which is maybe a more well-defined scope than the entire viral infection field, and say what is needed. As John said, what do we need in terms of models, in terms of modeling technology, in terms of data, and what are the important problems that get addressed. And then we need to find a way to meld those together. And as John suggested, maybe that could be a paper that gets published. It could be a collection of white papers, certainly. We could make a proposal for funding of different aspects of it. So those are all things that come out. So that's one of the, and maybe the first task. And the other task, of course, is that this group as a whole needs to catalyze research. You know, for example, last week we had a really wonderful discussion of various questions that came up based on the talk we heard. And I think that led to a number of good ideas of what one could do, different people who could do them. And so that's the other, maybe most important part of this working group, is to catalyze new research. Now, of course, a lot of that is sort of serendipity. It just, people get talking and stuff happens. But we might want to think about, you know, what's a good organized way to make this happen? Because as John also mentioned, if you have 130 people who meet virtually once in a while, it's difficult to really create a community and to create a space where people can share ideas. So I can put those things down on paper and make a sort of a proposed charge for the different subgroups and for the working group as a whole. But I hope that this is a discussion that we can have collectively and formulate a good objective for this group. And that's about all I want to say at the moment about subgroups and about the charge for the individual subgroups. But we should relatively quickly come to a point where we have a tentative list and get moving on it. So sometime, you know, by early next week, maybe, if you want to contribute to this, please do so as soon as possible. Thank you.
Okay, thank you, Reinhard. Are there, I know we're closing in on our end of time very quickly. There were a lot of questions after the mini seminar, so it took longer than perhaps we expected, but the conversation is a good one to have. Again, suggestions for how to work on the Slack would be appreciated. These ideas about organization would be appreciated. Anybody who has suggestions for things they want to be put on the agenda for next week, please let us know. Suggestions for speakers. There was a request that people might give us a one-page personal introduction that we could post on the website and the Slack. If people are willing to do that, that takes effort, I understand. There is a shareable document with subgroups and leads. And there also was an email sent out which allowed you to put yourself up as a group member or as a lead. Please do sign up for the subgroups. There is a generic Google Doc for comments and suggestions in general, the one that Jacob asked for. Jim, could you possibly just share these two links in the chat while we're talking? And those would be the main things. I can open it up for the last few minutes if people would like to talk about any of these issues openly while we're here together. As some people will drop off, but we certainly can talk for a few minutes about these issues if people have suggestions. So please feel free to express your opinions and ideas on how to handle these things. And you don't need to raise your hand. You can unmute yourself if you like.
So if you don't want to lead a subgroup, but you want to be part of it, is there a sheet to sign up? Because I think the Google only asks for leads.
So there should have been a questionnaire that was sent to everyone who had expressed an interest, allowing them to put their names down for any subgroups that they wanted to be members of, and also with a separate tick box for leads. Jim, if you could share the link to that questionnaire again in the chat, that would be very helpful.
I'll send an email to everybody when the meeting's over. It's a lot easier to capture it that way. It's already in the chat. I can put it in the chat again, but I will send the Google form lets you be a member of a group or volunteer as a lead of a group, and you can do either one. So
Thank you,
hopefully that'll get us the information we need.
Okay, I know we've hit our hour. If there are any other comments or suggestions to make, we have a few minutes if you'd like to talk. Otherwise, we hope we'll see everybody next week. I think this idea, I agree. There was another seconding of the question on the research topics. One of the main goals of this working group is to connect people. And so the point of having these mini seminars is to discuss research opportunities. The hope is there's serendipity and that people will find connections that they might not have found otherwise. The seminars are recorded and will be made available publicly. So if you have people you think should listen to these talks, please, the links will be made available on the IMAG MSM website, and you'll be able to watch them if people are interested. I know the world is pretty saturated with video conferences, so we don't necessarily need many more, but for some people that may be useful. That's why we asked people to try to keep their talks short as a sort of a teaser to get people interested. Any other comments or suggestions before we break?
Yeah, following any of the talks, if anybody wants to have a mini Zoom to talk to the speaker about that, outside of this meeting, it would be really nice to hear you say that during the meeting, to say, hey, I have more questions, and could we have a sidebar meeting? We're kind of trying to replicate what you might do if you had conferences where you would go off and have lunch together and pursue topics, and we're not together. But on the other hand, it's a lot easier to convene a Zoom meeting to drill into questions. And the main thing, I think, is just keeping momentum. I counted, I think, 32 people on this meeting today out of about 130 who signed up. That's absolutely amazing. And so ideas for just keeping the momentum going is really welcome.
Any final thoughts before we break? No? Well, thank you everybody for joining us. We should have 2 good speakers next week. We will send out, if you don't get any announcement on the sign up for the working group, please let Jim Sluka in particular know. If you have suggestions for additional members for the working group, please send our invitation on to them. We would love to have more people join us. And thank you all, and we'll be talking by email beforehand, and otherwise we'll see you next week.